Chapter 16, Part 7, Cell-Mediated Immune Response. So the cell-mediated immune responses are going against intracellular pathogens and your body cells that are abnormal. They're doing something that they shouldn't be doing. Sometimes that's cancer. Sometimes they are virus infected. So those would count as intracellular pathogens. Sometimes there's protozoa, there's viruses that can be intracellular. There are also bacteria that can be intracellular. So the cell-mediated immune responses work against things inside of cells and broken cells. The T cells, in particular the cytotoxic T cells, well, the T cells in general are the cells referred to in cell-mediated. These are immune responses that are done by cells. Which cells? T cells. We have cytotoxic T cells, and their clones function to find certain kinds of pathogens. So these start out in the lymphoid tissues, and we'll get a process, the steps involved in getting these cytotoxic T cells activated and ready to go. So first we have antigen presentation, where they detect that the antigen is present. The T cells differentiate, helper T cells differentiate. We get clonal expansion, and then we'll get self-stimulation, which isn't what it sounds like, I promise. So here we see a dendritic cell. It's an antigen-presenting cell, so here's its MHC class 1 with the epitope that it's put here. This is the T cell, the cytotoxic T cell, that is inactive. Here's the T cell receptor that's recognizing the epitope, and here is its co-receptor, CD8, to say, okay, you're the right MHC, okay, and here's the epitope, and if the epitope should not be here. We've already screened out the T cells that recognize self things, so this epitope is not self. This does not belong here. This T cell becomes activated and undergoes clonal expansion, which is exactly what it sounds like. The cell does mitosis, 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 and it makes a bunch of active cytotoxic T cells. It also tucks away some memory cells. We'll talk about memory cells in a bit as well. And so we get these active cytotoxic T cells that respond to different kinds of things, but there's also self-stimulation here. So the T cells are making cytokines that they then respond to their own self-produced cytokines. And so we get active, aggressive T cells going out to find where did this epitope come from, and they're going to kill whatever cell's making it. So the other part of this response is over here with the MHC class 2. Again, we're going to have a T helper cell that recognizes its epitope. When that happens, it differentiates and becomes another T helper type 1 cell that begins secreting factors to help the other T cells, the cytotoxic T cells, doing clonal expansion. So this is like a growth factor. It's going to stimulate expansion, stimulate these cells to go through mitosis. So we have now a big army of active T cells, and we've activated some helper cells as well. We can see this with a virus-infected cell as well. Here's our class 1 MHC protein. Here's our T cell with the CD8 to detect and make sure that this is the right kind of MHC class 1. Again, this is the protein that we're checking for before we do a transplant. We want the donor cells to have the right kind of MHC class 1 protein, so that the T cells that recognize with their CD8. So we're looking for these proteins when we're doing transplants. So this is all to just make sure that this cell belongs here and that they're making things that are okay. When it's a viral epitope, this T cell, this cytotoxic T cell, goes through that process that we just described in the previous. It activates and we go through clonal expansion. Once the cytotoxic T cells, the clones are produced and they're active, Those are cytotoxic T cells, and they have two different ways that they kill their target cells. They have the perforin granzyme pathway, and then they have the CD95 pathway. The perforin granzyme pathway, they make a protein that kills target cells. With the CD95, they target a particular glycoprotein on the cell's surface. So here is, in this panel, what we see with viral infected cells. Sometimes they use this mechanism. They make a protein called perforin. They secrete it. When this T cell finds a virus-infected cell and it has an epitope of something that should not be there, right? Which is because it fits into the antigen binding site. This cell releases a bunch of this perforin onto, and they're touching, right? These are two proteins on the surface. So these two cells are really close together. These perforin proteins perforate the plasma membrane. They punch holes in it. They assemble these complexes that make pores. They make little holes. And then this T cell begins releasing granzyme. And granzyme enters through the perforin holes, and they activate apoptosis enzymes. These are enzymes, every one of your cells has these enzymes in them. They almost never get activated, but when they do, within 20 minutes, the chromosome of that cell has been shredded. There's teensy fragments of DNA. Proteins are cut into tiny little pieces. Ribosomes aren't functioning. Nothing is working, and the cell is dying. So they cause the cell to do apoptosis, and hopefully they do it before the virus completes its life cycle and can assemble new virus progeny. Another mechanism that cytotoxic T cells sometimes use achieves the same goal, but in a different mechanism. So again, it relies on first having this interaction here with the MHC and the epitope being presented, triggering this T cell. And then the T cell will bring over, they have a CD95L, that's L for ligand. The other cell, the virus-infected cell, if it has CD95, which is a protein that has an enzymatic portion on the inside. If CD95L binds to CD95, then CD95 becomes active. And the enzymatic portion, the enzyme portion of that, what that does is it activates the apoptosis enzymes. And so it begins that apoptosis process. And again, within 20 minutes, this target cell is dead. And again, this happens with virus-infected cells. These can also happen with cancer cells. Cancer cells are often making strange proteins that aren't normal. Their genomes are often very strange, so they're weird in a lot of different ways besides just growing out of control. And so that gives us some opportunity for our immune system to find them and detect them and kill them. Now, previously I mentioned the memory T cells. Some of those activated T cells become memory cells. They can persist for months or even years in the lymphoid tissues. And so these cells just sort of hang out in there. They aren't really doing much. There's maybe screening fluids as they move through, right, as the lymph moves through the lymphoid tissues that these guys are in. They kind of screen and wait for their epitope to appear again. If you have a memory cell to something, that means that you have seen it. It's been inside your body. That's why you have a memory cell for it. And if you've seen something once, you might see it again. You might not, but you might see it again. So the memory cells are there just in case you see it again. And they wait. And if the epitope does turn up again, they can reactivate, and they will go through clonal expansion again. And it's a much more quick response than the first time you had the exposure. And so these memory T cells are a good way to have very fast response to a second exposure, to after you've already had something, already been exposed. This is, by the way, the function, the purpose of vaccines is getting a memory response. The T cells and the B cells both do memory cells. And the thing with the memory cells is once you have them, that means that you can get exposed again. This is why we want immunity, right? This is why we want to be able to say, oh, are you making antibodies? Because your memory B cells recognize and are responding and are ready to respond. The memory B cells and T cells are what mean that when you get exposed a few months later, a few years later, you won't get sick. So the memory response is faster and more effective, and usually if you have a memory already in place, then you don't even know that you got a second exposure or a third or a fifth or a 27th. So they're very effective, and you just never know that that was ever even a thing. You don't have any symptoms or signs of any second or third infection at all. So T cells need to be regulated to make sure that they don't respond accidentally to autoantigens. They've already gone through the clonal deletion process, but we still want to make sure that we don't have any slip through. So they really require multiple signals from that antigen-presenting cell before they really respond to the antigen. And then there's also regulatory T cells that sort of calm down the cytotoxic T cell activity over a longer period of time to kind of calm them down and get them back into sort of inactive state again. In the next section, we're going to talk about the antibody immune response.